All right. So as you trickle in, I'll sort of remind you about where we were in our exploration of the immune system yesterday. We're still on point 1, diversity in immune repertoires. We sort of understood that what we need the immune system to do is not recognize self, recognize the pathogens, and fit in an animal. And we talked about the generation process, which is called VDJ recombination, where you randomly combine one of the templates from the gene templates for the 3 parts that make up each chain of the receptor, V, D, and J. You edit the DNA, you take one, and then you randomly delete and insert nucleotides at the junction between the V and the D and the D and the J. Okay? Everybody remembers that? Okay, good. And you do it for 2 chains. So now the name of the game, and where we left off, is in that case, what is the probability of generating any given sequence? In other words, if you give me a random string of letters chosen from A, T, C, G, the 4 nucleotides, can I tell you what's the probability that that string was a T cell or B cell receptor that was made in your body? Yes? Everybody happy with that? And so what we're going to do, and we talked about it yesterday too, is use these out-of-frame sequences. So when we go in and do the sequencing of actual receptors in you, remember some of them will not make valid receptors. For example, they'll have a stop codon somewhere. But they're in our data, and we're going to use those. Remember the one from one parent, the other from this other parent business? Yeah. So we're using the ones in which the second one worked and was expressed and functional, but the first one wasn't, but is in the cell. And for those of you that are sequencing aficionados here, I told you how this works at the DNA level. I can tell you that even if you sequence mRNA, although the law is that, well, if it's not functional, it should be silenced. If you look in, you'll see mRNA in lesser amounts than DNA, but you still see it in sequencing. So even if you do mRNA sequencing, which is technically easier, you're still going to see it. Okay? The wonders of biology we can exploit. All right, so name of the game, if I call my sequence, so sigma is now, some sequence of letters. I'm going to completely make this up, and hopefully I don't write the stop codon into it, right? So it's some sequence of letters that comes out of a sequencer, and we want to establish the probability with which this sequence sigma was generated. So this is my data, sigma 1, sigma 2 up to sigma n. And what I'm going to do is, okay, so first of all, I told you about this word game that you could play to take 2 random words, delete, and so on. Hopefully that convinces you that if I see a given sequence, because of this inserting and deleting business, there is no way I can deterministically say how this sequence was made. Is that clear to everyone? Right? Because I can delete and insert essentially anything I want here. Right? So I can't say for sure that's how it was made. So what I'm going to do is I'm going to do it probabilistically. For each sequence that I see, I make a list, a library of all possible scenarios, by which I mean V gene, D gene, J gene, number of insertions and deletions, that are consistent with this sequence. Okay? So I only colored one of them, but all of them are the same idea, but they're just, so maybe I'll make it explicit. Okay. Wrong color. Maybe let's give this one less. Right, so to make it explicit, I could have chosen, the V has to be consistent with here, but as I said, some of them are similar. So maybe here I choose V9 and here V8. Here I choose D1, D2 and here D1, and so on. And let me choose the same J just to show that they can be the same. But then I can have a different number of the insertions and deletions here to make it work. Okay? I can imagine, just trust me. It works like that. As I said, I could show you a concrete example. You can play it with words. But basically, I come up with all the possible ways of making this sequence. Okay? And I do it for all sequences. Then, so then I have a model which I'm going to say is my recombination model, which basically describes this Wij, which is a model for how likely each of these scenarios is. And for that model, I need the probability of using a given V, D, and J gene, the probability of deletions in V given a given V gene, the probability of insertions in the VD junction, probability of insertions in the DJ junction, deletions, and so on. You get the idea, right? Yes? Of the indices? Yeah. The first index is the sequence index that I'm trying to represent. The second index is my guess index. Okay? So to calculate these weights, I need to know these probabilities. Where do I get them from? Well, I get them from data. So I make an initial guess. It can be anything. And I calculate the mean probability of using, of using a V gene given this set of weights. The mean probability of using D and J, and I'll explain to you in a second why I do that. The mean probability of, well, basically all of these elements. Okay, so since I wrote it like that here, let me write it like this here. I'll explain this in a minute. Okay, and so on and so on and so on. All the elements I need to plug into here, and then I use these to recalculate the weights. Everybody agrees now I have everything I need to recalculate these weights? I have the elements, I have the model.
How do you know that you have all the possible
I don't. Okay, so what I do is I make a very, very long list, but within reason, which is basically limited by some mixture of common sense and computational feasibility. And in practice, when the probabilities start going below something like 10 to the minus, I don't remember what it is, but 50, we stop. Okay? I'll get back to how these probabilities scale, but basically, okay, I can tell you now. So the more insertions you add, the less likely it is, because each insertion is random. So once you go above some number of insertions, it becomes very unlikely that you've, say, had 50 insertions. So we stop in that way, but we have about an order of 100 scenarios for each sequence. So it's not a small number. Yeah.
Sorry, I'm not sure I follow the logic. So you propose the Wij?
I guess, yeah, whatever, take them to be random.
But then is this a consistent check?
Yeah, this is then it's a self-consistent loop,
Okay.
which I haven't said yet. So then we do this, okay? And then we iterate this until convergence. And there's a reason it's bound to converge. Can somebody tell me why? I used the word yesterday and some of you claimed you know what it is. If I now call this theta as of my parameter, and I call this pseudo likelihood, does anybody recognize it? I'm doing something in 2 steps. I'm updating, I'm calculating my weight, so I'm calculating my pseudo likelihood using my prior guess of the parameters of the model, and then I'm using this pseudo likelihood to update the parameters of my model. Yeah, it's expectation maximization done on your fingers with no math. Well, the math's in the board. Okay? I'm basically, so because it corresponds to, for those of you that know what expectation maximization is a maximum likelihood procedure that has hidden variables. Okay, so instead of just having in maximum likelihood, I have the probability of my data given my parameters. Here I have the probability of my data given my parameters and some set of hidden variables. Okay? I'm summing over the hidden variables, which in my case are the scenarios. So I call them scenarios, and these are hidden variables. And then and it's a 2-state procedure where you fix your guess of the parameters, you update your pseudo likelihood, then you take that pseudo likelihood to minimize it to update your parameters. And you do this until convergence, and since we know it's a well-known algorithm that some mathematicians or computer scientists have proven to converge, we know this iterative procedure will converge. But you don't need to know any of that to understand the procedure, hopefully, right? You guess, you update, you have a model, you recalculate, you update, you recalculate, you update, recalculate, and you trust me, it'll converge. Okay? So at the end of the day, we get these weights, and now we want to get the probability, right. So we want to get P-gen of sigma, which is a sum over all the scenarios that give you sigma of these recombinations of S. And again, you get it by maximizing likelihood and this is the number of data sequences. And this is a model. And these are model parameters. Okay. Yeah.
So we're finding the weights that make this data maximally likely?
No, yeah, well, no, we're finding the parameters. Let me write it out. Okay? We find parameters of the model that make the likelihood most likely, and then from the model we calculate the weights. The weights are really the hidden states. Yeah.
What is the difference of this technique basically? Because I was say you just minimize the Lagrangian. What makes it special?
There's nothing. It's just there's nothing special about it. It's just maximizing log likelihood. It's just maximizing log likelihood, but in a Bayesian way. I mean, you sum over. You don't know directly how this depends on the data, so you add the hidden states. If hidden states doesn't mean anything to you, you just basically add this all the possible scenarios. Right? Basically what we're saying is, I don't know whether it was W13 or W116 that actually in reality produced this sequence. So what I'm going to do is I'm going to sum over all of them. Right? So very good question. Thank you. I was waiting for that question. Yeah.
I don't understand
Yeah.
actually why there are some conditions.
Exactly. Good. That's why I said I'm going to have to do some explaining. I'll just write out so I'm maximizing log likelihood over these parameters, which, okay, I don't want to rewrite, but like P, V, P, DJ, etc. So which means I'm maximizing the log sum over scenarios of P of data scenarios parameters. Okay? To answer that question. Now, okay, the key is sort of here. Where did I just wrote this down. Why did I factorize this? Why didn't I factorize this? Okay, so this is a model which is the most general. So there's two ways of thinking about it. Either you're a biologist or either you're a physicist. First of all, we were physicists, meaning we knew nothing about the process. So what we did is we wrote down the most general model that is still consistent with the data. Meaning we tried to factorize different everything, and then if it didn't work, if it didn't converge, then we said, okay, our model is wrong. And then we came up with this. Now, had we been a little bit smarter and actually managed to make our way through the textbook, we would have guessed this. Because what is known, so basically what this tells you is that the choice of V is made independently from the choice of D and J, but that the choice of D and J is not made independently. Right? That's what factorization, that this is independent from that. So why is that? Well, it's independent, that's sort of what you would assume, right? An enzyme comes, makes the choice, and then moves on. In fact, the way that on the genome, the genes are organized, the green, blue, is the following. D1, J1 to J6, D2, J7 to J12. So that means that if I choose D2, remember I'm cutting DNA, I can only choose G7 to G12. Whereas if I choose D1, I can choose, but that's why the choice is not independent, because of the genome architecture. And everything else, choosing it to be independent is consistent, of course, except for the deletions on V depend on the deletions on each gene depend on what you're actually deleting, and so on. So in a way, it's just the most common sense, most general, most factorizable model consistent with the known biology. Yeah. No. These genes are pretty long. I mean, it is possible for D, and it does happen for D. But for V and J, they're too long. But for D, it definitely happens. And what's more, D is so short that basically from after this in our lives, we stopped worrying about D, because you realize that whatever you see, it's very hard to tell apart whether it was the original D or whether it was just a few nucleotides from D that got randomly inserted and looked like D. So when people tell you it's this D gene, they basically can't be telling. There's no way of really knowing that. So that's a very good question. All right, anybody else? Everything else clear? We have a model, right? So now you give me this random, you write down your favorite set of, you have a question? I have 3 dots because I'm lazy, and if I need to write down everything, it'll be, But yeah, you have the probability of inserting DJ. Okay, you do deleting D, deleting J. You can also have a probability of sequencing errors in this likelihood, if you so please.
And what about the pieces, these random pieces?
Yeah, so that's this. That's the probability of insertions, and in this probability of insertions, we can add also the probability. There's also terms for the probability of what gets inserted, and there we also considered whether these are independent nucleotides, or there's like triplet, whether there's some correlation in what gets inserted. It turns out that the probability of the next base pair you insert depends only on the previous one. So a Markov model of insertions works. But basically, you're getting the idea. I don't want to, right? Okay. So now you can do this, write this down on a piece of paper. And if you want, then you can go to a website, which is called sos@statphys@dns.fr, and type that in and find out what the probability of generating that sequence is. Yeah. Yeah, very good question. That's where we're getting. Okay. So my line here. Ah, okay, good. Good. All right, so that means I have this much space that I can use. Okay, so what do we get out of it? I mean, first of all, what we get out of it is distributions for all of these. I don't really want to bore you with them. There's different, so first of all, it's not homogeneous. The gene usage is not homogeneous. There's different things. You probably don't, I'll answer that in words. I can maybe show you as a side note somewhere. Let me just do it here before I get to that. So just to give you an idea, the probability of insertions as a number of insertions looks like this. This is about 20 here. This is for humans. Okay, this is what we get from the model. So this is now, this is a parameter of our model. If we didn't do this, this summation over all possible scenarios, but we would just take the most likely one, the one that has the largest weight, this probability would look like this. Okay, so it has this spurious peak at 0, which isn't there. So that means that doing this fact that we really cannot say which one is the most likely one is important, and we do have to do this summation over these hidden states. But anyway, this is what they look like. You get graphs like that. There's a lot of parameters here. But at the end of the day, let's get to the biology. So, probability density. This is log base 10 of pgen of these sequences. Okay, I'm gonna this is about -8. This is about -4. This is about -25. Okay? These are human T cells, for example, right? We can do it for everything. Okay, so what is this? I calculate this probability of generation of sequences that I see in my data. Imagine I take blood from you. I take a unique set of your T cells, and for each of these T cells, I calculate the probability of generation. Okay? Then I make a histogram. I ask how many T cells did you have that had a probability of generation that's 10 to the minus 10. I put a dot here, 10 to the minus 12, 10 to the minus 15, and so on. I make a histogram like that. Is it clear what I'm doing? Okay. This is what it looks like. Now, say I've taken Massimo, and now I take Caroline. And now I take Etienne. And now I take Alex. And now I take Alessandro. And now I take every one of you in the room. And now I take 1,000 different people. Okay? This is the variance of all of us. So it means we're all very reproducible. Okay? It doesn't matter how much soy milk you drink, what you do with your life, your probability of generation of sequences is the same. And with Caroline in the room, I should not say this, but it's all dumb biochemistry. Right? It's just our enzymes at work. Okay? Yeah. Yeah, very good. Okay, what does -8 mean? So remember, we have 10 to the 9 unique. Okay? So what does 10 to the -8 mean, or 10 to the -4 mean? 10 to the -4 means each one of us will likely produce this sequence. What does 10 to the -25 mean? You're probably going to produce it yourself. Right? So what that means is we have a set of sequences that are very common in the population and ones that are very rare. And immunologists have these concepts and words, and they talk about public sequences and private sequences. They don't talk, well, now they're starting to talk about it based on this, but the idea is basically what historically, if they would find the same T cell in all of us, well, even in 2 people, they'd get super excited, right? Oh, it must mean something. It must be there for a reason. It must be recognizing something. What this shows is this T cells that we're going to find in all of us just by pure chance, for no reason. They just have a high probability of generation. There's also some sequences that are specific to you, which means that if we sample all of you, I'll be able to recognize each one of you just from your blood sample, which means you shouldn't kill anyone because, you know. Okay? And when I say that, this actually differentiates identical twins. Why? We had this question yesterday. I just don't remember whether it was during the talk or after the talk. Yes, it was after the talk. Somebody came up to me and asked, do you pass anything, do you pass the T cells or B cells to your children? You don't. You pass on the machinery You pass on these genes, and your kids make them by themselves from scratch. That means your kids will not have the same repertoire you have. And that means two identical twins will not share basically much more than you and me. I'm assuming none of you are my siblings. Well, even if you were my siblings, we wouldn't share any more than if you're not my siblings, basically, right? Okay, so who has an identical twin? See, there's always someone in the room. Okay, so I can tell you, when you were, did you have a joint placenta? Yeah, so if you had a joint placenta, then as I said, you start producing these things in utero, and in fact, as in embryos don't do insertions. So they just put things together and they don't do insertions. So then if you have a joint placenta, you have one bloodstream, so you actually share the ones that you produce as an embryo. But then the moment you're born, you start producing your own ones. So by now, you're probably not sharing that much more with your sister than with anybody else in the room. But there is still some, since T-cell clones, not cells, but clones have a lifetime of about 40 years, you still have some remnant of your embryonic life in your repertoire. As do all of us, but for the rest of us it doesn't matter since we have nobody to share it with. Okay? So anyway, point number one, it's very universal. Remember here we're talking about unique and generation. So at that level, we're all the same. And this is really data from over 1,000 real life people. And even so, there's no differences. Somebody asked me about how different the genes are, and I told them about polymorphisms. So in fact, the gene choice is the most different part. That's the thing that how often you use a given gene is the thing that matters, it differs the most. This thing is incredibly reproducible. Like if you look at error bars on it, it's like this. However, as we'll see later, gene choice matters very little to the overall distribution. Okay, I talked about some common, some universality. Yes? That's a very good question. So that's a question that I will not answer. I know that was what the answer to that partly lies in the optimal immune system. So I'll just tell you the answer. Well, honestly, we don't know, but if we ask how would you optimally design an immune system, because of cross-reactivity, so remember the balls, it doesn't really matter which receptor you have, whether you have that receptor or a slightly different receptor, as long as they recognize roughly the same things. And since there are so many different ways of recognizing the same thing, it doesn't seem to matter. So basically, this setup seems to work. Questions? Yes? Yeah, well, it's not self-anti. Your proteins get passed on, yes. I mean, your genes coding for your proteins. Remember, you have two parents. Don't forget about the other parent. Don't overemphasize the mother.
Yeah, but my question is, during timing selection, since the T cells get selected based on if they bind to cells or not, then won't there be some similarity between parents?
Yeah, that's a very good question. So first of all, again, I assume you have a mother and a father, given how old you are. So don't underestimate the role of your father in your life. Probably roughly half of your MHCs come from him. So, you're not the same as your mother. right? You're a mixture of the two. And so probably what you're presenting, like, that's why it's also so hard. Like, a parent can't usually give a transplant to the kid for this reason, right? So you retrain it on this combination that you've been dealt. But there is some similarity. Basically, what you're asking is whether the MHC matters in the training. It does matter. It's actually this is a very complex question. We've looked a lot at this. Basically, can we separate out the signature from the MHC from the signature from the peptide? It's even more complicated. So there's two types of T cells, CD4 and CD8. And they have different set of MHCs that present to them. And I'm going to mix, there's a nano technique that I keep mixing up. That doesn't matter, you won't remember anyway. But anyway, there's two sets, they're completely disjoint. And so you would imagine that you should be able to tell apart T cells that recognize one set of MHCs and T cells that recognize the other set of MHCs. It's really, really hard. Like, there is a minute difference, and most models can't do it. So, but that's an open problem. Okay. So now we know how to generate stuff. We know things are heterogeneous. We know it's very universal. But we basically have numbers for generating anything, for anyone. So what happens next? After we've generated sequences, what happens to them? Selection, exactly. Okay, so remember thymic selection. This is the question of, do you bind to anything at all? Do you bind too strongly to self? So now we're going to use our in-frame reads. So the functional ones, the ones we assume are functional. This is an assumption. To learn something about selection. So first of all, here we were working at the nucleotide level. Selection happens at the amino acid level, exactly. So I need to get my PGen and get it into amino acids. So I need to sum over all the nucleotides. So this is the sum I had before. I now need to also sum over all the combination of nucleotides that give me a given amino acid sequence. So realistically, there's about 2 to 4 codons at each site. Not all amino acids happen equally. There's about 15 amino acids at each site out of the 20, not all the same, but just to give you an order of magnitude. So this sum is actually about 15 to the 2 to 4. It's a huge sum. So I can't do it exactly. So what I'm going to do is I'm going to do dynamic programming, which I'm not going to explain to you. But I'm going to tell you what it is as a physicist, which is basically transfer matrices. So everybody solved the 1D Ising model in their life? Okay, well, no. But if those of you that haven't solved the 1D Ising model, have you seen dynamic programming? And some of you haven't seen either. It doesn't matter, okay? There's a trick. There's a trick where you go back and forth and you manage to do the sum by breaking it up into many steps. Okay? So either you know what this means or there's a computational trick. Doesn't matter for what I'm going to say. Okay. So after that, I can define P post selection as Pgen as the probability of, so the probability of seeing a given sequence after selection is the probability that it was generated times a selection factor, which I will write this way since I'm a physicist, but if you don't like that, you can just call it Q. Okay? It's a selection factor. So basically what I'm saying is for each sequence, how much does it get modified? It's like my F, right? But now my F at the actual level of data. Okay. And so we can recalculate, we can calculate it, we can take your in-frame sequences, and I can add it to this graph. And so now P post will look like this. Okay? It shifted slightly towards higher probabilities. So that tells you that somehow the way this machinery works is that sequences that will be selected during phi-mix selection have a higher probability of generation. I can tell you another tidbit of data, which is that during phi-mix selection, you keep about 15% of sequences but only 2% of diversity. And I'll tell you what that means later. Latest coming in 2 minutes. Okay. But I want to... I don't remember. I don't, from visually, not much. May probably some. Honestly, I mean, yes. I mean, there must be a little, but it's not like it's the, because the key here is we're still at the level of unique sequences. I'll get away from unique in a second, but at the, you know, there is a little bit more, but I don't remember the numbers. Okay, so let me, maybe I should tell you, I should explain this now, and then I'll get back to my last point. So I'm gonna delete, should I erase this? I can erase this. Okay, so diversity. Finally, we're getting to the end of this. Okay, so what I can do now is I can make another plot, which will be my clone frequency. Well, my really, TCR frequency versus rank. Okay, so what I do is I take my TCR sequences, TCR1, TCR2, TCR3, and I order them by the count. So I first put the one, I'm making numbers up, but you'll get the idea. I look at how they count in the data, and then I give them a rank. So I put the most common one, as one. Or actually, why don't I not? That's one way of doing it. I could also order them by the pigeon. Let me order them by the pigeon. So I take the most likely one, which will still be like like 10 to the minus 4, 10 to the minus 5. Okay. 1, 2, 3. You get the idea, right? I basically order them by the pigeon. Sorry? I can't hear you. T-cell receptor, yeah. Sorry, thank you. Okay, so this is rank 1, 2, 3. And then I make a distribution of the log of pgen versus the rank. And it'll look like this. Okay? The reason I was putting counts is I can do the same thing with counts. I can normalize the counts of what I see in the data and plot it versus rank. And I'll also see something that looks like this. Okay. So what do these numbers tell us? This is the most common one, right? How likely is the most common one? This is the total number of sequences, right? Because all I'm doing by rank is I'm just counting them. Or in this case, this would be the total number of sequences. So this would be the repertoire size, as we've been calling it. This is the most common clone. Okay. This is the total number of unique. So that could also be a measure of diversity. Okay. In fact, here there's an interesting point. This has slope 1. This is data. Right? Just to be clear. Okay. So we're back to the diversity question, and we're trying to figure out how to quantify diversity. So one quantification of diversity are basically these distributions. That's the full quantification of diversity. I mean, in fact, it looks a bit like this. And then there's a sampling cutoff in all of them. So from the data, it'll go, sorry, maybe I should draw it. It goes, go like this. Right. This would be, okay. Okay, so one way we know how to quantify entropy is, diversity is entropy, right? So that's what I'm going to do. Okay. So this is Shannon entropy. Well, I'm going to sum. I've been using sigma, so let me use sigma log base 2 because we want bits. Okay. So this is a general measure of diversity where I have a distribution over some species, in my case, of my different receptors, or my probabilities can be pgen, and I sum over the species, and I get a measure. It's one measure. I wanted to actually spend part of a lecture talking about different measures of entropy, but I won't. so this is one version of a class of diversity measures, information theoretic diversion measures, based on what's called Renyi entropy, which is defined as this. This is just a side note. Okay. Use the same notation. P to the beta. Okay. So if you take beta to 1, you recover the Shannon entropy. I can do it, I cannot do it, doesn't matter. I just want to focus on this because we don't have much time. Okay, it's... Yeah. No. So it can be, this is just a general definition. For what I'll do in a second, it'll be P gen sigma, right? Because I want to know, my first question is, what is the diversity of the generation process? And for that, I'll take P sigma to be P gen sigma. Okay? Okay. Has everybody seen this before? Yes? Even the people who don't know what transfer matrices are? Have a general idea? Okay, it's a diversity measure. The basic things to know is that if for a uniform distribution where P sigma is 1 over m for all sigma, S is minus log base 2 bits. What's a bit? One bit is the outcome of flipping an equally likely coin, in the sense that you have two possibilities and they're both equally likely. So if you have two equally likely states, that's 1 bit. And generally, it's bounded between this. Okay? So if there's only one state that is occupied and everything else is 0, then the entropy is 0. There's no diversity. That's what it means, right? No diversity, one solution. And this is equally likely. So max diversity. And it has units, it has bits, so it's all good. Again, it's a special version of Renyi entropy, where other special versions include the Simpson index, if you've ever heard of that, which is an overlap measure. Do you want me to tell you about that? You don't care. Let's see. Let's get back to this. Which is related to the Gini-Simpson index, which maybe some of you have heard about. Anybody? Yes. Yeah, Gini-Simpson, it's the inequality index when they look at countries and who has larger inequalities and stuff like that. But for our purposes, let's just stick to this. And let me... Can I erase this? Maybe this was a mistake to erase this, but whatever. It's hopefully ingrained in your minds by now. So, we're answering this question now. For the generation process, what is the diversity? So, okay. So, generation at the level of nucleotide sequences, beta and alpha. is 47 bits, and this is 30 bits. So you see that beta has a higher diversity, which doesn't surprise us. At the level of the recombination process itself, it's even larger. Why? Because not everything you recombine will actually be functional. And now the breakup into VJ insertions and deletion. Okay. Before we go there, so just what does these numbers mean? So that means roughly 47 equally likely scenarios. So if I take that to 2 to the 47, I get of the order of 10 to the 14, I think. I don't remember. Equally likely ways of making that sequence. Now, this distribution tells you that it's anything but equally likely. Equally likely is a bound. Basically, what this tells you is the diversity is infinite. Okay? And I'll make a point. Why don't I make this point now? In here. So, what does that mean? How does this graph? Okay. So, you can ask yourself, has every T cell that can be made, has it been made by humanity? Right? It's a fair question. If I'm telling you something's infinite, it's a fair question. So I'll plot the number of people alive on Earth today, which is 10 to the 10th, versus the log, this is also a log, of course, log of the distinct T cells. And so I said we've sampled around 1,000 people. So that curve, starting from 1, looks something like this. But we have a model that can extrapolate. Okay, so what do we expect from this graph if humanity has made every possible T cell receptor? It'll saturate. Okay. So the model goes above the sampled and continues. So, okay, it continues into this graph. Where we are today, we're nowhere near saturation. That's the diversity. So this is for one chain. If I add this and that, I get 77. If I turn that into equally likely sequences, that's around 10 to the 64. I don't like this number because biologists misinterpret it. Basically, it's huge. There's nothing else to say. The capacity of this system to generate numbers is huge. Receptors is huge. Okay, so there we are. Okay, the only good news for humanity probably at this point. We still can generate new T cells and B cells. Yeah. Yeah. This is data. The difference. Yeah. So when we sampled until about 1,000, that's point 1. The second point, why is there a difference? Because when we sample these humans, we don't bleed them to death. We're nice people. Okay. Right. Okay. So now, so this is the answer. This is the answer, the generation level. Now we need to still break this up into contributions from VJ, insertions, and deletions. So the question now is, which of these processes contributes most to the diversity? Who votes for gene choice? VJ. Anybody? Yeah. So remember the scenario is you choose your gene, you do your deletions, and you do your insertions. I'm going to break up the entropy into these three processes. The question is, which one of them is the one that contributes most to the diversity? Who votes for the choice of the genes? 1, 2, okay. Two votes here. Who votes for insertions? Okay, a lot. And who votes for deletions? Okay. So I'd say around 5 and a lot. My non-binary. Okay. So the answers. 9 bits for genes, 11 bits here. 15 bits and 28 bits and 6. So both of you were right because it depends on the chain, but basically the genes don't matter. Okay, so all of you that are worried about passing stuff on to your offspring, it's not so important. Okay, genes don't matter. Yes. Yeah. No, surely they've been selected for, I'm being facetious here, right? Genes matter. First of all, they provide the scaffold, right? They've clearly been selected upon. If I replace these genes by random genes, so actually what's interesting, people use this process now in synthetic biology to generate diversity. But, they will not generate B-cell. It's an interesting question if you just, if you engineered some V genes and, try some that you think are better in some sense, for example, taking them from as a final product of affinity maturation, where you have all these mutations, and, use that in it, what would happen? That's a good question. Nobody knows. They didn't want to fund that project when we proposed. Yeah.
So how can they?
Because the genes are templated and insertions and deletions are random.
And very short.
They can, I erased it. On average, it's 5. The peak, the median is 5, but they go up to 20 with, 15 insertions still can have significant probability. Yeah. But it's very, very random, right? At each position, you have 4 choices. So even if I put 5 in, that's 4 to the 5.
But they don't affect the receptor so much.
They do. That's the point. This is right. Remember my crappy drawing from yesterday? Okay, this is where they go. Is at the tip here, which is the part that interacts. These are these loopy hands, and so they're very important for the receptor. And again, it's really just because of the that basically you have some number of genes that are templated, and you don't change them. Whereas here you have two knobs. One is whether I add another insertion, and the second knob is what do I add. And I mean, you should be surprised. And like you know, immunologists didn't like this result, right? They also, I mean, a lot of them were very confused by this result, that you can just generate stuff by chance that is the same. Yes? Can you start again? Just. Yeah. Yeah, that's during thymic selection. That's what's being tested. Oh, but whether every V with every J will form a structure that folds? Yeah, no, right. So, I mean, that is one of the reasons they're important, the genes. I mean, they're also, you know, they're not completely, they are relevant for interactions. So, okay, so this region is called CDR3, which means complementary determining region 3. There's also, I don't know where it is, it's somewhere here. In the V gene, there's two other regions which are called CDR1 and CDR2. They're also known to be very important for direct interactions. So it's not like they don't matter, right? Just for the diversity that templates it. Whereas now the insertions and deletions do something that's completely non-templated, and that's why it adds diversity. Okay. So I said this, so let me go back to my, to this here for a second. All right. Yeah, okay, I started this because I wanted to explain this result. And I actually don't have the number, so let me, and I just erased everything. Let me look this up. What do I have here? I have the answer for your diversity question. Gene P post, yes, it's slightly bigger, but it's not a huge difference. Looking at it this morning, so I know I have it here. Right, okay, so post-thymic selection, I mean, the numbers don't really matter, but here, what did I have here? It's 44, 38, okay. So this number goes down to about 38 bits. Okay? And so based on this I'm giving you this number that you keep 15% of the sequences. I didn't show you how, but if you take the ratio of those two numbers, maybe slightly different versions of the numbers, you find out that you pick about 2% of diversity. Okay? So this is basically consistent with this, that you remove sequences during thymic selection, but again, you don't do it completely randomly. There's some laws to how you do that. Okay, and so last thing, which we've already said, is sharing. So so now we know what's the probability with which we generate the sequence. We know what's the probability with which we select this sequence, right? So we know what's the probability of seeing this sequence after selection. Everybody agrees? Right? These are numbers that we just multiply. So if I want to calculate the number of shared sequences between you and me, what do I do? What's the probability that if I sequence you and I sequence me and I look in a naive repertoires, that I'll find exactly the same sequence? What had to happen? Yeah.
We get the probability using the concatenation of both of our repertoires minus each one.
It's a simpler way. Okay. What had to happen is you had to generate this sequence and it had to pass Fimec selection, and I had to generate this sequence and it had to pass Fimec selection. You and I are independent entities. We agree? So it's just P post squared of that summed over that times, to get the number, times the sizes of our repertoires. Everybody agrees? It's random, right? Everything is random, both the generation, the selection, and the fact that you and I are independent. I don't think we have to discuss that, right? Right? We're all random machines, yes. Yeah. Yeah. But I mean, it depends on the individual, but again, as Tarek asked, so maybe I should put this up, at the level of unique sequences, I mean, there's so much I can do in this, but since this bit of chalk is a little bit thinner than the other one, my point is not getting, this is a bit fatter than that, but not much. So we can build a universal model for everybody, and we can build separate models, and actually, so I can do P post you times P post me. It won't make a difference. In fact, let's build a universal model for everybody and do that, because it's part of the point I want to make. Okay, so then the number of shared sequences between you and 10,000 of your closest friends, or not closest friends, is just going to be n1 to n,000 sum over a p post to 1,000. Agreed? Okay. So let's draw it. Log of number of shared sequences, number of people sharing. 1, 10, 1,000. And let me first draw the model prediction, which looks like this. And now let me put data on this. Okay? basically we can predict how many sequences, again at the level of unique, you share. I mean, we can also predict it at the level of non-unique. This number per se, of course, will depend on sampling. It'll depend on this. But, you know, we can correct, we can renormalize. There's sort of, you know, there's a lot that goes into this. Basically, we're now able to say how many sequences anyone will share. And back to these concepts of private and public, what this shows is that there's no such notion as really public. There's a degree of publicness. Because whether you share a sequence with one other person or 10,000 other people, it's like a completely different setup. Okay? Yes? Dynamic programming thing. I wrote Q as e to the e sigma over z. It's an energy. That's why I wrote this for physicists. Sorry, whereas I wrote this for you. Okay? It's just a factor. It's, okay, what is E sigma? E sigma is just, the whole thing is like a correction factor, and this is where the model goes in, right? So why do we write it as E of sigma? Because then when we try to fit models, there's different ways we can do it. If we write E of sigma, we will be fitting what are called an exponential family of models. And what's the simplest exponential family? Is an independent, I'm running out of letters, HI. So I can write a model like this, where I assume selection acts independently on each amino acid. That's the simplest kind of model I can write. We call it a linear model. I can write a model like this that then has Jij terms, which means that there's interactions between amino acids in selection. I can add third order terms, or I can forget about all of this, and I can just say that I have a deep neural network that learns E of sigma. And then it has all possible nonlinear terms. So a model like this we've called Sonia. And a model like this we've called Sonia double N. Okay? And you can play this game, you can learn different models. And honestly, this doesn't do, I mean, it does slightly better and captures some effects, like you asked the difference between DM8C. So we threw that at it. This is good enough for all practical purposes, such as that, and for many things. This does pick up some small things, but of course this requires huge amounts of data. So if I'm trying to then I can also use all this machinery to look at different subsets, like to look at T cells that are responding specifically to some perturbation. And since I need tons of data for this, then that's hard. But then I can use transfer learning approaches, which means I first learn a model, I learn a model like this on generally what makes a good TCR on some subset where I have a lot of data. So for example, I learn just the PPOS model, and then I learn a model like this on this specific subset. I can play games. And we get into zoology of that. But this, basically what I want to tell you is, even at the level of your naive repertoire, you're not, at the level of unique sequences, you're not so special. Okay, you're predicted with well-parameterized models through chance. Now, the thing we didn't talk about, that I sort of tried to highlight here, is that, as we said, that when T cells respond, they proliferate, we get to a situation where we have clones of different frequencies. Okay? And that's what's shown here, is if I now do the same thing, I do TCR1, TCR2, TCR3, and then I look at my data for counts. That's why I was coming up with these crazy numbers. I don't know, it's super hard to write random numbers, right? Anyway, and I give them ranks, 1, 2, 3. That's what makes this. And we do differ. I mean, okay, so thing to be said, I said this has slope 1. So I can do it for you, I can do it for me, I can do it for everybody. We all have slope 1. At this level, this distribution, again, looks very, very similar. However, which T cell will be largest than you is specific to you. Okay? And that's back to the question we had of, does it matter, you know, that we don't generate the same TCRs? No, it doesn't matter, because you'll end up with this, but just with different identities. Okay, so I'm nearly out of time. So I'm not going to tell you about optimal repertoires. I can refer you to the paper, which is I'll refer you to 2 papers, which I actually don't, as always, remember the dates of. When was this published? 2014. And for I think it's 2023. So this is traveling waves. They're pretty clear, so you can just have a read. What else I wanted to say something else. I wanted to write. Okay, but I do want to end with a quiz question. So in this optimal immune repertoires, we work in this phase space that I introduced. Cross-reactivity is important. We assume there's a fixed set of pathogens out there. Okay, so just imagine you have a distribution in this space of bad guys. I'll give you the punchline. So let me make this space, remember shape space that I drew as a box with the balls and everything. I'm going to make it one-dimensional to make it easier. And in it, I'm going to give you a distribution, a random distribution of pathogens. Okay, this is now the question is, how do I place my immune receptors in such a way as to maximally cover the pathogen space? I have a finite set of receptors. I can't just do this, right? I can't draw a high line somewhere, right? So I want to match this as closely as possible. What do I do? First of all, question, should you and I have the same optimal distribution of receptors or not? No, good, right? Why not? Because of everything we said and because of cross-reactivity. Now, cross-reactivity is key here, so it doesn't matter exactly where I place my receptor. Where should I place my receptors? how should I place them? Should I just draw a uniform line somewhere? Should I distribute them somewhere? Where the peaks are? Exactly where the peaks are? Yeah. Okay. So what I should do, you know, see, very good. I put delta functions in a way that I cover space they're not the same height, so this one will be maybe slightly less. This one will be maybe slightly more. Just didn't draw it well, okay? Slightly. Okay? They are delta functions placed in such a way as to completely cover space, because remember, cross-reactivity does this for us, right? So that if I look at coverage from these receptors, I'm going to have close to unival coverage of the pathogens. Okay? And these are mine. And these may be yours. Okay, it's getting busy, but you get the idea. Yeah, the local concentration. It should somehow integrate what's under the... Yeah. Okay? That's the punchline of how I got there and how the derivation goes. And if you want to learn about constraint optimization and KKT constraints, read the paper. All right. I want to say two more things. One, I'd like to thank the organizers for inviting me. This was really, really much more fun than I thought it would be. So I'd like to thank all of you for that. Hopefully you enjoyed it too. And last thing, we do have postdoc positions open specifically to work on immune repertoires and co-evolution with viruses and differences between wild mice and real mice and stuff like that. So if anybody's interested, very data-driven projects, come talk to me. All right, I'm done.